Hello, Science 30, and welcome to the rest of our lesson on DNA after the cell cycle. Today we're going to look at life in 4 letters, the structure and function of DNA, and replication. So here on the screen, you're going to see four very important letters, G, C, A, and T. These are what make up your DNA. So let's talk about what that is. DNA is a twisted ladder or a spiral staircase. It's called a double helix. It twists around and it kind of looks like a ladder because it's connected from one side to the other in the center by what looks like rungs. We say DNA is made out of 2 complementary strands, meaning they're not identical, but they match each other. So rather than identical twins, think fraternal twins, born at the same time, but don't match each other, but complement each other. The backbone of the DNA is made from some sugars called deoxyribose sugar and a phosphate molecule that are connected together. And then through the center, from one backbone to the other, we have these nitrogen bases, adenine, guanine, cytosine, and thymine. They make up the center of DNA and are paired up in a specific pattern. So there are rules when you pair up DNA. So in DNA, we refer to a sugar, a phosphate, and a nitrogen piece as a nucleotide. And the rules are as follows, that anytime you have an A, it pairs with a T and vice versa. If you had a T, it would pair with an A. If you have a C, it pairs with a G and vice versa. If you have a G, it pairs with a C. The reason for that has to do with the fact that DNA has different sizes as their nucleotides. So you can see here, I said that adenine and guanine are purines. What that means is they're much bigger structures, as you can see from the picture here. Okay, so therefore they want to pair with a small structure, thymine and cytosine. So what happens is this bigger structure of G will always pair with this smaller structure of C, and A will always pair with T. Now you're probably thinking, well, why can't T pair with G and C pair with A if it's big and small? And the reason for that is the way that they are designed is that C and G are going to always make triple bonds and A and T are going to make double bonds. So that means when they bond together, they only have certain spaces to bond certain types of times. So you can only ever pair C and G and A and T together. That's going to lead you into the part in your notes where you're going to do a bit of a review and learn about how DNA is paired up. You're going to skip down from there and go down to what is the main functions of DNA. So the basic functions or the main functions of DNA, we're going to start with number 1 as transcription and translation. In very simple terms, that just means protein manufacturing. Our DNA has little tiny things on it called genes that are like the recipe book to building all the proteins that make up you. Proteins function in specific ways. They're catalysts or enzymes. They stabilize your body like muscles. They respond to stimuli like in neurons, and they carry nutrients around your body. Okay, so we're going to talk more and more about proteins, and you're going to see the importance of them as we go through and learn about transcription and translation. The next really important thing for DNA to do is through inheritance. So why do some offspring not look like their parents and some do? Like, what's going on there? Why do you not look identical to your sibling? And the reason that is because DNA is inherited, and when it does, the proteins have slight differences to them. So genes that code for proteins are slightly different, and therefore you could get a gene from your parent or an allele from your parent that has codes for blue eyes and one that codes for brown eyes, and it might look a little bit different for your siblings. So inheritance is a really unique function of DNA, passing on information from parents to offspring. The last one is replication, and we're going to write down some really key steps for replication, but essentially replication is DNA is inherited from one generation to the next, but in order to do that, we have to replicate it so we can pass it on from parent to offspring. So what is replication? It's essentially the unzipping of your chromosomes or your genes, okay? So here's a good example of what that would look like. If this is DNA, here's our backbone is the blue part, and all of these are the rungs of DNA. Replication is unzipping and then making a copy of it. So where you have replication in your notes, we're going to define it, and then we're going to write the 4 steps down. So DNA replication is the process in which the DNA molecule is duplicated before a cell divides. That's all you need to write down, what replication is. It's when DNA molecules are duplicated before the cell divides. Now what happens is our 2 complementary DNA strands, okay, get unzipped, and then we build new complementary strands on either side. This is really, really important so our cells can divide and pass on information. This process is called semi-conservative because we use a parent strand, one here, and then we build two new strands. So every time we build DNA, you have a parent strand and a new strand that stay together. So what are the processes of DNA replication? Well, the first process is that we need to separate our strands. So the two DNA strands unwind and separate, breaking our hydrogen bonds between all of the A, T, C's, and G's. Then what needs to happen is we need to complement the strands. So we take and we pair all of the open nucleotides with their complementary pair, following the rules of A's and T's stay together and C's and G's stay together. Now, there are enzymes and pieces that help with this process, such as DNA polymerase, helicase, and ligase, and we're going to talk a little bit about those in class. Then what happens is we form two new strands. So we have, this is that semi-conservative strand that I was talking about. Your two new strands are built from an original strand and the new strands because we use the old strand as a building block. Now that we have two identical copies of DNA strands in a cell, we're now ready for cell division. So that's the 4 steps for cell replication that you need to write into your notes. As I said, we're going to add a few more terms to help us understand who's helping out in the process, but here are the steps I would like you to write down underneath that definition. If you have any questions, please let me know.